Today in biology class, I'm going to present the DNA and RNA detailed structures. Here, DNA, that is deoxyribonucleic acid, forms a double helix with deoxyribose sugar. The base is adenine, guanine, cytosine, and thymine, and is the primary genetic material. RNA, that is ribonucleic acid, is typically a single strand using ribose sugar bases adenine, guanine, cytosine, and uracil instead of thymine, and folds into complex shapes to fulfill various functions. Both are nucleic acids composed of nucleotide monomers, which consist of a phosphate group, a 5 carbon sugar, and a nitrogenous base. The same thing could be seen in terms of structure here as I have presented. That is cytosine, guanine, adenine, uracil for RNA, and in case of DNA, adenine, guanine, cytosine, and thymine is present. So here you can see on the left side that is RNA, which is single strand, and then right side that is DNA, which is double strand. Then here you also could see in DNA that is base and the pairings, while you cannot see this thing in RNA case. And you can see few terms, for example, nucleobases, that is cytosine, guanine, adenine, and then uracil or thymine. While here you can see the another term that is base pair, that means adenine binds with thymine and cytosine with guanine by using triple hydrogen bond. So in case of cytosine and guanine, you could see the triple hydrogen bonds, while adenine and thymine binds with double hydrogen bonds. And this is the kind of significant difference, main difference, which you need to know in one word. Then the DNA structure, it is double helix. DNA consists of two polynucleotide strands that bind around each other to form a double helix. Sugar, the 5 carbon sugar in DNA is deoxyribose, which has one fewer oxygen atom than ribose. So this is another difference which you need to remember, that it is deoxyribose which has one fewer or lesser oxygen atom with respect to ribose which is present in RNA. Nitrogenous base, DNA uses four nitrogenous bases: adenine, guanine, cytosine, and thymine. Base pairing, the bases form hydrogen bonds with a complementary base on the opposite strand. Here A pairs with D, that is adenine with thymine, and guanine pairs with cytosine, that means G pairs with C. Backbone, the sugar phosphate backbone forms the outer structure of the helix, as you can see here, this one. So this is what you can see, the backbone. Now, RNA structure is single-stranded here. Unlike DNA's double helix, RNA is usually a single strand of nucleotides. Sugar, the 5 carbon sugar in RNA is ribose. Nitrogenous bases are here adenine, guanine, cytosine, and uracil, which is in the place of thymine here. Folding, because it is single stranded, RNA can fold back on itself and form complex three-dimensional shapes such as clover leaf structure. Now, similarities between DNAs and RNAs. In terms of nucleotides, both DNAs and RNAs are polymers made from nucleotide monomers. Phosphate groups, if you want to understand here the similarity, each nucleotide contains a phosphate group. Nitrogenous bases, they share three nitrogenous bases: adenine, guanine, and cytosine. So these are the three main similarities which you can observe between DNA and RNA. Now, if you want to understand now the difference at the level of sugar and base, then here in DNA you can see deoxyribose, and in case of RNA it is ribose. And the difference you could observe here, you need to try to find what could be the differences. For example, here you can see H group, here OH group is more present in RNA. And then in case of bases, here thymine is present while there is uracil, And the difference between thymine and uracil, you need to understand with this one here, as you can see. Now, the DNA structure, if you want to understand, as I mentioned, that there will be binding possibilities between adenine and thymine between the two strands, not within the strands. So, and the bonds will be two hydrogen bonds in case of adenine and thymine. While in case of guanine and cytosines, there are three hydrogen bonds between guanine and cytosine. And these bonds are represented here with the dashed lines. And if you want to see their structure with the bonds, they may look like this. Here you can see the double bonds between adenine and thymine and triple bonds between guanine and cytosines. There are also a structure size difference also. So the guanine, cytosine, adenine, thymine, they are somehow different also in terms of molecular weights. Now, if you want to see how they are attached with each other, you can see here, for example, adenine, guanine, cytosine, thymines, they are. So here there are two types of bases. If you classify, then there are two types of bases. One is purine and another is pyrimidine. Purine are adenine and guanine, while pyrimidine, here you can see the thymine group. Now, what is thymine here? This you can see here. So the purine and pyrimidines are the two fundamental types of nitrogenous bases that make up DNA and RNA, with purines featuring a two-ring structure, adenine and guanine, and pyrimidines have a single ring structure, that is cytosine, thymine, and uracil. A purine consists of a six-membered rings fused to a five-membered ring, while a pyrimidine is a single six-numbered ring. These distinct structures are crucial for DNA's double helix, as a purine always pairs with a pyrimidine, maintaining a constant width. Now, you can see the detailed structure here. A purine here, heterocyclic aromatic organic compound consisting of a pyrimidine ring fused to an imidazole ring. Here you can see pyrimidine is a heterocyclic aromatic organic compound similar to benzene and pyridine, containing two nitrogen atoms at a position of 1 and 3 of the six-membered rings. And here you can see what in terms of structure. Now you can see with another way of structure for purines and pyrimidines. Now the purines here, adenine and guanine, you can see there are two rings, and pyrimidines have single ring in case of cytosine, uracil, and thymine. Now the same purines and pyrimidines could be seen here. If you are trying to write some kind of objective type questions, this kind of figures could help you to understand, like 6 plus 5 membered rings, purines. Here you can see. And then here, six-membered rings, pyrimidines. All the three structures, cytosine, thymine, and uracil, here they have six-membered rings. While in case of purines, you have 6 plus 5 membered rings. And purines are heterocyclic aromatic compound with a pyrimidine ring fused with an imidazole ring. While pyrimidines are heterocyclic aromatic compounds with a pyrimidine ring which has nitrogen atoms at the position of 1 and 3 positions. Now, the same thing could be presented in a different way. So here this is thymine, and thymine will look in details in the structure which you can see here clearly. While here uracil, they are slightly different. Now adenine, guanine, and cytosine you can see here. So there are different way of remembering or memorizing the structures. And in terms of compare, if you want to understand how they could look like if it is DNA double helix structure, in terms of structure, if you want to see, 5 prime to 3 prime, and 3 prime to 5 prime. This is how their structure could be represented or drawn. Now, DNA and RNA, if you want to go for complete detailed comparative study, then DNA and RNA differ in sugar level. So in the sugar, and where one has deoxyribose, another has ribose. Base comparison like thymine and uracil. Then structure, one has double helix, another is single strand. And primary function also, long-term genetic storage versus protein synthesis and gene regulation. DNA serves as the stable genetic blueprint, while RNA acts as a temporary messenger carrying instructions for a protein production. Now the table is summarized presentation of comparison of the key features. So here in case of DNA, deoxyribonucleic acid, sugar is deoxyribose. In RNA, it is ribose. In bases, adenine, guanine, cytosine, thymine. While in case of RNA, it is adenine, guanine, cytosine, and uracil. A structure mostly double helix structure, while here typically single strand structure is for RNA. Function, long-term storage of genetic information, the cell's permanent blueprint, that is DNA, while RNA carries the instructions for protein synthesis and other cellular processes, a temporary copy of genetic information. Stability, DNA is more stable and suitable for long-term storage, while RNA is less stable and suitable for its short-term roles. Location, DNA is mostly found primarily in the nucleus, while RNA is present in the nucleus and the cytoplasm. Now, the same thing you can see. The detailed comparison functions. DNA replicates and stores genetic information. It is a blueprint for all the genetic information content within an organism. Now, the RNA converts the genetic information content within DNA to a format used to build proteins and then moves it to the ribosomal protein factories. A structured DNA consists of double strands arranged in a double helix. These strands are made up of subunits called nucleotides. Each nucleotide contains a phosphate, a 5-carbon sugar molecule, and a nitrogenous base. RNA only has one strand, but like DNA is made up of nucleotides, the RNA strands are shorter than DNA strands. RNA sometimes forms a secondary double helix structure, but only intermittently. Now, the length, DNA is much longer polymer than RNA. A chromosome, for example, is a single long DNA molecule which would be several centimeters in length when unraveled, while RNA molecules are variable in length but much shorter than long DNA polymers. A large RNA molecule might only be a few thousand bases pairs long. The sugar in DNA is deoxyribose sugar, which contains one less hydroxyl group than the RNA's ribose, while RNA contains ribose sugar molecule without the hydroxyl modification of deoxyribose. Bases, the bases are in DNA, adenine, guanine, cytosine, and thymine, while in RNA it is adenine, guanine, cytosine, and uracil. Base pairs, there is pairing of the bases, and that is here adenine with thymine by 2 hydrogen bonds, cytosine with guanine by 3 hydrogen bonds. Here, adenine in case of RNA, adenine and uracil could pair, cytosine and guanine could pair. The bonds will be for adenine and uracil, 2 hydrogen bonds, while cytosine and guanine will remain the same 3 hydrogen bonds. Location, DNA is found in the nucleus with a small amount of DNA also present in mitochondria. That is called mitochondrial DNA. RNA forms in the nucleolus and then moves to a specialized regions of the cytoplasm depending on the type of RNA formed. Reactivity due to its deoxyribose sugar, which contains one less oxygen containing hydroxyl group. DNA is a more stable molecule than RNA, which is useful for a molecule which has the task of keeping genetic information safe. RNA containing a ribose sugar is more reactive than a DNA and is not stable in alkaline conditions. RNA is larger helical groups mean it is more easily subject to attack by enzymes. Ultraviolet sensitivity, DNA is vulnerable to damage by UV light or ultraviolet light, while RNA is more resistant to damage from UV light than DNA. Now, what are the key differences which you need to summarize and keep in your mind between DNA and RNA? you need to think about function, sugar, basis, structure, and the location. And this thing we have discussed a lot about the bases, sugars, and the so and the details. Now, what are the three types of RNAs? mRNAs, transfer RNA, and rRNAs. So, mRNA is messenger RNA, tRNA, transfer RNA, and rRNA is a component of the ribosome factory itself, without which protein function would not occur. Now, the sugar, both DNA and RNA are built with the sugar, which you know, deoxy and ribose. So, deoxyribose and ribose sugars. Now, bases, you are clearly uh, like understood that what are the bases present in DNA and the RNA. Now the location different. DNA is located in the nucleus, while RNA, depending on the type of RNA, the location will vary. Now there are different types of DNAs, that is unusual types of DNAs and RNAs. Here, ZDNA, while the structure of DNA you will see above and in the biology normally textbook, has the right-handed helix DNA molecule with left-handed helices also exist. These are also known as ZDNA. Canonical classical DNA is called bDNA. Now ZDNA molecules are thinner, 18 angstrom wide as opposed to 20 angstrom wide than bDNA. Now have a different repeating unit, two base pairs as opposed to one, have different twist angles between the bases. ZDNA is thought to play a role in regulating gene expression and may be produced in the wake of DNA processing enzymes like DNA polymerase. So ADNA, you can see in general, the structure is somehow like here, identified at the same time as bDNA, and aDNA is an alternative DNA structure that often appears when the molecule is dehydrated. Many crystal structure of DNA are in the form of aDNA form. It has shorter structure with different numbers of base pairs per turn and tilt than bDNA. aDNA's biological relevance has been greatly expanded on the recent years, and it is now recognized that aDNA is involved in many roles, such as binding to DNA enzymes and then protection from the damages. Now, these are the different forms of DNAs, aDNA, bDNA, and zDNA. These things you can clearly see in how they are different. So, this is what you need to understand. And now, another form of DNA is triplex DNA. The triplex helix DNA structure can form when certain nucleobases, pyrimidine or pyridine, occupy the major groups in conventional bDNA. This can happen naturally or as part of intentional DNA modifying strategies for the research purposes. So, triplex forming oligonucleotides, TFOs, can bind conventional two-stranded DNA, which can help guide agents that are used to modify DNA to a specific genomic locations. HDNA is an endogenous and triple-stranded DNA molecule that increases mutation of the genome. So here you can see how the triplex DNA could look like. Now then there is another DNA, that is dsRNA, double-stranded RNA, as and it's so this is this is about the RNA form. Double-stranded RNA, dsRNA, is most commonly found as the genomic basis of many plant, animal, and human viruses. These include Rio, Viridi, and the rotaviruses, which are responsible for the diseases like gastroenteritis. And dsRNA molecules are potent immunogens. They activate the immune system, which then cut the dsDNA as a protective mechanism. The discovery of the protein machinery that permits the reaction led to the development of gene silencing RNAi technology, which won in 20